See here, gluconeogenesis. Gluco means glucose, neo means new place, genesis means synthesis. So basically, glucose is coming from a carbohydrate source, either from starch or from glycogen. That is the universal fact we all know. But suppose if glucose is synthesized from a non-carbohydrate source, like suppose if glucose is synthesized from a protein or if glucose is synthesized from a lipid, then you call it as gluconeogenesis. Okay, synthesis of glucose from non-carbohydrate compounds is called gluconeogenesis. Okay, so for which you have to learn cycle and you should show the sources of proteins and also like glycerol, which is now going into the cycle of gluconeogenesis. So first I am going to take you to this cycle beta. This is actually citric acid cycle in short, because this is all reverse way which is explained here, and you have to write the points also in reverse direction. So first thing is, this is citric acid cycle. So we know like pyruvate is the last step of glycolysis in aerobic conditions. So from pyruvate, you have oxaloacetate, alpha ketoglutarate and all. Now, so at the level of pyruvate, pyruvate can be synthesized from the amino acids. Number one, alanine, glycine, serine, cysteine, threonine and tryptophan. These are the amino acids which can form pyruvate and which can lead to citric acid cycle. Okay. Second thing is oxaloacetate. So oxaloacetate can be synthesized from the amino acid aspartate and aspargine. Then alpha ketoglutarate can be synthesized from arginine, glutamate, glutamine, histidine and proline. Number 3. Number 4, succinyl CoA. Succinyl CoA can be synthesized from isoleucine, methionine, valine. Okay, number 5 is fumarate. Fumarate can be synthesized from phenylalanine and tyrosine. So these are all the things what you are finding as like the agents which are amino acids which can enter into citric acid cycle. Now you should take this cycle in reverse direction better. So now see here, from pyruvate you can see this is the step phosphoenol pyruvate which we see in glycolysis. Okay, that means glucose can be synthesized from the amino acids. Phosphoenol pyruvate can be converted to, now these are all reverse steps. This is reverse cycle of glycolysis. You need not memorize anything. It is glycolysis only. You are finding arrow in two directions. Okay, from phosphoenol pyruvate, reversible arrow you can see. So you are finding 2-phosphoglycerate, then 3-phosphoglycerate, 1,3-bisphosphoglycerate, glyceraldehyde 3-phosphate. From here, fructose 1,6-bisphosphate, fructose 6-phosphate, glucose 6-phosphate. Finally, glucose is formed. Okay, so that means from the various amino acids, intermediates of citric acid cycles are produced, and from those intermediates, pyruvate is produced. From series of steps back, glucose is synthesized. Okay, that means from proteins, glucose can be synthesized. That's what is gluconeogenesis. This is the cycle of gluconeogenesis. Now, from fats also glucose can be synthesized, for which you have to know this. We know glycerol, triacyl glycerol is nothing but fat, cholesterol. So from glycerol, you are finding now the cycle has to be taken in reverse steps. Glycerol to glycerol 3-phosphate, glycerol 3-phosphate to dihydroxyacetone phosphate, from here to fructose 1,6-bisphosphate, and finally it will lead to the formation of glucose. So that's what is better. So amino acids can be converted to glucose, and also fat also can be converted to glucose. So that's what you have to write for this answer. Describe the roles and responsibilities of physician in communicating diagnosis of thalassemia. Okay. What is thalassemia, beta? We have alpha thalassemia, we have beta thalassemia. In alpha thalassemia also, we have 5 different types, isn't it? So the 5th type, like we have 4 genes which can carry for this, no? Hydrops fetalis, we have thalassemia just trait, and finally you will see the last step where the person will not survive only. Okay, so that is hydrops fetalis, where person will die. So that means that is alpha thalassemia. In beta thalassemia, we have beta thalassemia major and beta thalassemia minor. So in this condition, we have to give counseling to the parent. Like if all four genes are defective, basically the baby will die immediately after birth. But if it is alpha thalassemia where only one or two genes are defective, still only it is the alpha thalassemia trait. So where if we give blood transfusions, the survival rate of the child is better. Even in case of beta thalassemia minor, there also if we give at least blood transfusion once in a month, the risk of death we can lower. So in such case, but blood transfusion where you will get very good blood and where you will see the normal hemoglobin is present. So for such people, there are generally thalassemia clinics. So we should give address to the patients like where these thalassemia clinics are situated, which are free of cost, working free of cost. Okay, so we have many such centers located in Hyderabad. Okay, in all the other district hospitals also we have such arrangement of blood transfusion for the patients who are suffering with thalassemias. So we have to give counseling to the parents not to worry much, and we have to connect them to good government hospitals where they can get treatment free of cost and there will not be any burden for the patient and also for the parents. Okay, so that's what is counseling. Actually, it's a good question. Explain the synthesis of ketone. Ketone bodies, clinical significance, nothing but ketonemia, ketonuria, ketogenesis. This is enumerate the biomolecules of extracellular matrix. Okay, biomolecules of extracellular. This we will see better. Meanwhile, you try to revise the cycle of ketogenesis. That is ketone body formation and ketone body breakdown. We have collagen fibers, type 2, type 3, type 4 collagen fibers. Okay. So these collagen fibers will be seen in all connective tissue. It will be found in basement membrane, it will be found in bone, it will be found in muscle, everywhere. Whatever protein part we have in our connective tissue, that is nothing but collagen fibers. Okay. So basically we have four different types of collagen fibers. Okay. So let me open and then you can see also. Got this question like collagen fibers, isn't it? So collagen fibers are nothing but protein fibers. First, collagen is most abundant protein in mammals. So I told you everywhere, whether it is bone protein or muscle protein or blood protein or whatever, even in the basement membrane, everywhere we have collagen only. Okay. So collagen is the major component of connective tissue. Collagen gives strength, support, and shape to the tissue. Okay. Now, once you see the types of collagen fibers, I told you we have in Roman numbers. Basically, when we discuss in anatomy, basically we talk only about four: type 1, type 2, type 3, and type 4. But he says like we have many types of collagen fibers. At least 19 different types of collagen fibers are seen. When you see the structure of collagen fibers, better you learn this picture better. Diagrammatic structure of collagen and a fibril. See, you have to draw all these like wavy lines. These lines, straight lines, which are not continuous. These are all collagen fibrils. So each collagen molecule will be made up of many collagen fibrils. One collagen molecule, it is shown here, it is measuring around 300 nanometers. And these collagen fibers, they are actually coiling. Like you see for DNA, double helical structure, like that you will see two, three, four coils will be there, which are nothing but the collagen fibers which are coiling. So each collagen fibril, when you see, it is made up of alpha chain, it is polypeptide. Polypeptide in the sense it is made up of different types of amino acids. Like basically glycine is predominant type of amino acids. In between we may have different other types of amino acids. So just look at this structure while you read the structure of collagen. Okay, you need not do biosynthesis of collagen and all, that becomes too much. Okay, so anyhow, for now this question what you had, like biomolecules of extracellular matrix, you can elaborate glycosaminoglycans, then just give this list: collagen, elastin, fibronectin, and different types of extracellular matrix elements. Okay, then compare and contrast glut2 and 4. Let me check this answer. If anybody has this answer, they can share with us. See, group 2 and group 4. This is our 3 marks question, right? So, neurons like GLUT2. What is this? GLUT2 are found predominantly on. GLUT is nothing but glucose only, beta. See, glucose which is used for the purpose of energy by the neurons. It will be in the form of either 2 or 4. Let us see. See, GLUT2 are found predominantly on the basolateral side of the membranes of intestinal and kidney cells, as well as beta cells of pancreas, whereas GLUT4 are the glucose transporters found in muscle and adipose tissue. Okay, what from where they gave, we don't know also. Compare and contrast. So where do we find GLUT2? It is found in the membranes of intestinal and kidney cells and beta cells of pancreas and liver cells, whereas GLUT4, these are glucose transporters found in the muscle and adipose tissue. That you can write. So this question, next question is advantage of auto analyze. Maybe auto analyzer this is. This is also from your lab manual, this question, auto analyzer. Okay. I hope you should have seen when you went to your biochemistry lab an image I will show you, like auto analyzer, like see an instrument which looks like this. Okay. So the advantage is it is commonly used in soil testing, but we will see its use in biochemistry lab. Okay. So it is using the pale yellow supernatant, that is serum is used of centrifuge blood sample and also urine sample, and induces reaction using reagents to measure various components such as sugar, cholesterol, protein, enzyme, etc. So that means this analyzer, auto analyzer, what we use in biochemistry lab, it's an instrument which we can use to measure the levels of glucose, cholesterol, protein, enzyme, both in serum and also and in urine. That is the purpose of this auto analyzer. Okay. Okay. Reverse cholesterol transport. This we will see. Component of cell membrane. This we have seen Singer-Nicolson model. Bilipid layer studded with protein. Functions, it is semi-permeable. That means what? It is selectively permeable. Whatever nutrients and whatever minerals and whatever things which are useful to the cell, only those chemicals will be allowed to move across the membrane. Glucose will be allowed to enter into the cell membrane. Certain minerals like sodium, chloride, potassium, which are useful, they will be allowed to enter into the membrane. but there are certain harmful drugs which will not be allowed to cross the biological membrane, that is our cell membrane. So reverse cholesterol transport we will see better.
Ma'am, only semi-membrane.
Yes, cell membrane means that only, no, but what we have studied, Singer-Nicholson model, green and pink
Yes,
layer
ma'am.
I have shown you, no?
Yes, ma'am.
That only you have to write here. That is only the cell membrane. Components in the sense, it is made up of bilipid layer. Bilipid layer is shown in green color, and that pink
Hmm.
proteins, whatever are shown, those are proteins. That means bilipid layer studded with proteins is nothing but our cell membrane. And function is it is selectively permeable. Okay. So now he is asking us cholesterol, reverse cholesterol transport. It is only 3 marks question. No, that's why I'm sharing like this. So I hope you are seeing on the screen. So reverse cholesterol transport is a process by which cholesterol in non-hepatic tissues is transported back to liver via plasma components such as HDL along with ATP binding cassette transporters such as ABCA1, ABCG1, etc. What does it mean? See, basically cholesterol is here in this mechanism. It is transported back to liver from non-hepatic tissues. That means here picture is also shown. No, we will open this picture. So cholesterol is being transported from non-liver tissues. That means it is being transported from tissues like adrenal gland or some like intestines, from the stomach, from the kidney, from various other parts of the body. Now the cholesterol is being transported back to the liver. So that's what is called as reverse cholesterol transport. We will read once again. Process by which cholesterol in non-hepatic tissues is transported back to liver via blood. That means through blood circulation only. In the blood also we have specific components which are binding with cholesterol, like they are HDL along with ATP binding transporters, special transporters. The name of these transporters are ABCA1, ABCG1, etc. Okay. So that is reverse cholesterol transport. So remaining part of this paper, which is also 50 marks, we will do tomorrow better. Okay. So for next 3 days,